I think we can start. So again, welcome everybody to the ELSC Spring Seminar Series. Today we're hosting Dr. Eran Stark from the Faculty of Medicine in Tel Aviv University. Eran received his MD in 2006 from the Hebrew University, but he also an ELSC alumni, and he did his PhD with Moshe Abeles and graduated in 2008. He then did a postdoc at Rutgers NYU with Georgi Buzaki, and in 2015 he's a faculty at the medical school at Tel Aviv University. Eran research focused on the neural circuit level to understand processes such as short-term memory and spatial navigation. To this end, he uses high-density recording of neuronal activity with optogenetic tools to manipulate circuit at a single cell resolution. And I'm sure he's going to tell us all about this. So before we start, the usual announcement, we encourage questions during the talk. So if you want to ask a question, please feel free to do so. Just unmute yourself and go ahead. And of course, there's going to be time for questions at the end of the talk. So without further delay, Eran, the stage is all yours.
Okay, so thank you very much, Didi, for this invitation. And well, I hope you will enjoy the talk anyhow. So I will talk to you about this title, Error Correction and Reliability Timescale in Converging Cortical Networks. And before I start, I would just like to acknowledge and describe the contributions. So basically, all of what I will describe to you is work of Amir Levy. He did all of the data analysis and most of the figures, and almost everything that I will describe to you is his work. The actual data were recorded by these four people. Let me modify the pointer. So these are the students that recorded the data: Amir, Adas, Shirley, and Lidor. We have an animal colony which is managed by Ortal and with the help of Tamar and Noah in the past. Some of the probes that we are recording with are custom made by Fan Wu, a long-time collaborator who is now in the DBC. They make fantastic probes. I have no financial interest in this, but I encourage anybody to go and see this. And this is the funding. So the basic puzzle, perhaps, that I would like to address is the following. We are constantly exposed to inputs at all types, all of the time. And some of these inputs are constant, some of them are changing, some of them change slowly, some change rapidly. And clearly the inputs are processed along the nervous system. And there are sensors that sense the inputs. Some inputs are internal. I dream about something, I think about an idea, but some of them are external. And when there's an external input, it is rather amazing that the temporal precision of the inputs is preserved for multiple stages along the processing pathway. So this is an example from an experiment done about 25 years ago, in which Gabor patches were exposed to monkeys, and they recorded from MT, which is a higher level visual station, after the retina, after LGN, after V1. And in some of the cases, the stimulus was moving in one direction, the classical experiment, and in some cases it flickered back and forth at a frequency of several dozen of hertz, about 50 milliseconds. So it's not super fast, but it changed. And somehow the neurons in MT, or some of the neurons in MT, followed these flickers. So this is one example. Another example is from the Nelkin lab, where they recorded from cats auditory inputs. Again, the inputs were not fixed, they were constantly changing natural sounds, and they recorded from various parts of the auditory system, but somehow in A1, which is a relatively high, 4 synapses or so from the periphery, some of the neurons exhibited consistent and relatively precise spiking over multiple presentations of identical stimuli. So how can that happen? This is what I would like to address. As I said, multiple stations must be crossed, so it could be retina, LGN, V1, MT, it could be cochlea, inferior colliculus, MGN, and A1, whatever it is, or it can be multiple internal stations that an internal thought is processed along the way. But so the question is, how is the precision maintained? And we know from mathematics that when you process information, you can only lose it, right? You cannot add information by any signal processing or any relays. So you can only preserve information or morph it. So the question more formally is, how can information be maintained, or temporal information can be maintained over multiple synaptic interfaces? So this is the question. And then when one wants to ask this question or answer it, you need some kind of a reduction. So the reductionist approach that we chose to take is to focus on a single synaptic interface. So we're thinking about the brain as a multi-layered, recurrent system, but everything can be connected to everything. But you can always plot it as something like this, and we can focus on one interface. So this will not give us a general answer, but it will give us a partial answer. The other decision that we took is to use a wideband input. So specifically, we're using filtered Gaussian white noise, which is not our idea. People have used this almost 50 years ago at very different time scales. This is 2 seconds, and this is a very different preparation than what we are going to use. But the concept of using a wideband input allows us to look at various time scales and try to determine which time scale is relevant for or can be preserved in the processing. And the third decision that we made is to use an intact system. We want to use a freely moving animal, actual brain. We don't want to cut out part of the brain and look at the rest. We want to minimize the interaction, the damage. Of course, once we do anything to the animal, we're intervening with the system, but we would like to minimize that. So these are the basic decisions. The actual zoom-ins are, I will start with experiment in the cortex, in which we focus on the interface between pyramidal cells and PV immunoreactive interneurons. I will explain the experiments with filtered Gaussian white noise. This was used several times in the past. Maybe the most famous example is a later example in which brain slices were blocked completely. All the receptors, everything was blocked, and the single pyramidal cell was injected intracellularly and exhibited very precise responses. But we want to remove that. We want to use the free moving animal, the unblocked receptors, and we're not really focusing here on the generation of spikes. We're more interested in the transmission of information. And the third thing is to use an intact system, as I said, and we're using freely moving mice. We build crowns on their heads. These are Faraday cages in which we place high-density electro-optical devices that we build in the lab. This is a photograph, a micrograph of a dual-sided two-shank probe. Each side has 16 recording sites, 20 microns apart in the vertical dimension. So there's 64 sites overall. And we put on them optical fibers, and when we stimulate one fiber, we can generate, this is not a single cell effect, this is an effect of maybe 50 cells that are spiking, but it's localized to one shank, and there is basically minimal effect on the other shank, and vice versa. So this is the kind of technology that we use. Now, to quantify the time scale, we need to formalize two very important questions. We need to define what is a time scale that we are interested in. And when people talk about spike timing, sometimes it's not clear. So I want to make it as clear as I can. So the time scale of spike trains or spike timing can be measured with relation to several references, and I will relate to two references. One is the external input, that is the white noise that we will use. It can be anything else. It can be the bird chirp, it can be the flickering of the Gabor patches, it can be anything, but it's some external input. And the other reference that one can take is another spike train under similar conditions or as identical conditions as you want. And so that will be the equivalent of the first one will be the equivalent of how much do I understand what is somebody telling me. And the second definition or reference will be how much do I understand the same thing when I am told the same thing. It does not mean that I need to understand the meaning, right? But it means that the spike trains will be similar to each other during a certain condition. So I will name these two references by precision and reliability timescale. Okay. So how do we go about this mathematically? So the framework is the following. We use a frozen white noise. So in all of the experiments, it's exactly the same white noise. It changes the amplitude and the DC, but it doesn't change the shape. And we take a cell or a group of cells in the brain, and we apply these stimuli by optically by injecting light. So it's a photocurrent that we basically inject to the cell many times. We do not control for brain state. We allow the animal to do whatever it wants. We do not allow control for the position in space. We allow it to walk around in the cage or in the open field. So the animal is freely behaving. We record everything, of course, but we keep exactly the same stimulus many times. Now, once we do this, the neurons sometimes, some of the neurons, obviously not all of them, those that are closer to the light source spike. And they spike, this is an example of 10 spike trains from an example neuron. This is a real recording. And they look more or less like this. So it's not punctuate intracellular precise timing events, but it looks like this. So how do we go about this analytically? We aim to reconstruct the stimulus from the spike trains. We do this using cross-validation. We do tenfold cross-validation. We take 90% of the trials. From these trials, we build a Wiener filter. It's similar to the STA, that Bialek and Reike, the famous things that people use. But we normalize it or divide it basically by the autocorrelation of the spike trains during the stimuli. Using this filter, we can do two things. One, we can measure the timing of the peak of the filter relative to the spike. So this we call the time lag. And two, we can convolve this filter and reconstruct the stimulus from the 10% of the spike trains that we did not use to build it. And with this, we get a reconstruction. Now we do this for every single trial. So that's basically the framework for reconstructing the input from the spike trains. Now, once we do that, we can define precision. So how do we define precision? We take the reconstruction of every trial. Let's say this is trial I, and we compute the rank correlation at zero lag between that and the actual input. We call that QI, and we average that over all stimulus presentations. So this is Q, the reconstruction quality. And then we take the spike trains, every single spike, and we jitter it within an interval. So it's not an interval which is around the spike itself. It's what we call interval jitter. And after we do that to every single spike, we repeat the entire process, everything again. and we compute Q again, and then we jitter it at a slightly larger window, and etc., etc. So then we can build a graph which shows what is the reconstruction quality, Q or Rho, as a function of the half width of the jitter interval, delta. So the place where this is the original Q, and this is the Q after the reconstruction quality after jittering, and once this deviates sufficiently from the original Q, we mark this jitter window as the precision. So that's how we define precision. Next, we define reliability not based on this reconstruction. What we do for reliability, we take all of the spike trains, and for each spike train, we decide on a temporal window. This is an idea that Schreiber et al. Originated in 2003. What we do is exactly what they proposed. So we take every spike train, we convolve it with a Gaussian. For example, here a Gaussian which is very narrow, 0.4 milliseconds. And then we compute the cross correlation, or again, the correlation coefficient at zero lag between the two analog signals. So we go from a digital or point process representation to a continuous representation, not through the reconstruction method, which is data-based on a linear filter, but here it's based on a Gaussian. So it's a predetermined kernel. But what we do is we again change the filter size, or the kernel width, or the sigma of the Gaussian. And you can see that for a very narrow filter, the correlation or the reconstruction quality is so. And for a larger filter, it's higher, but for a yet larger filter, it's actually lower. So again, we build this curve of the reconstruction quality here again, or the correlation coefficient between the spike trains. Here again, it's not with the Wiener filter, but it's with a Gaussian kernel. And we do this for the same range of time windows, timescales. We call this sigma here. And we take the peak of this curve as the time scale of the reliability. Okay. Now, what was interesting was that this curve was thought to be monotonically increasing, but it turns out that if you do not keep the number of spikes constant, you actually allow the number of spikes to change, you can actually get these peaks. So once you do that, you can get a peak. These examples are fake examples, and to actually understand whether this framework works, we generated the fake data, synthetic data that we produced ourselves under exactly the assumptions that we want to test. So here you can see an example of punctuate event-like spike trains. So these have high precision and high or whatever short reliability timescale. And other spike trains, which were designed in the same manner, can show different effects. So for example, this spike train has a low precision, but has a peak in the reliability profile. So the spike trains are they're not very precisely related to the input, but they're similar to one another at a short time scale. And in this example, the precision is low and the reliability timescale is long. So we can actually dissociate between the precision. It can be high or poor. So short times for precision I will refer to as high, and long as poor. And the reliability timescale can be short or long. So when it's super short, I will refer to it as temporal coding, and when it's long, I will refer to it as rate coding. Of course, this is just names, but we can give numbers on this two-dimensional graph to each of the set of spike trains. And these things are not really, they're somewhat dependent on the firing rate, but they are not really dependent on the firing rate. You can have very high precision at a fixed or very low precision at the same reliability and vice versa. So that's the framework. Now, once we have this, we go to the brain and we look at what we see. So in what follows, I will focus on experiments in which we stimulated the pyramidal cells. So these are mice that express channelrhodopsin under a CAM kinase promoter, and we record all of the cells. Some of them will be pyramidal cells, some of them will be interneurons. Typically we record about 50 cells in a session. This is a particularly nice session with about 200 cells. Sometimes we have this. During this time, we can look at the spike trains of each neuron and relate them to the spike trains of the other neurons. And from this, we can generate a circuit map. Basically, using cross-correlation analysis, we can determine whether there are any pairs of cells in which after one cell spikes, so this is a pyramidal cell, whether there are any other cells that spike after it within a monosynaptic or very short timescale, and this specific interneuron spiked after about 2 milliseconds, and then its activity went back to baseline. So these are cases in which we would define the two cells are as presynaptic and postsynaptic, and using this type of circuit mapping, we can say, okay, these 4 cells which were recorded simultaneously make synaptic connections or spike transmit spikes at least. On that we can be sure mathematically at least to this interneuron or to this specific cell. So how do the actual spike trains look during the white noise simulation? So this is an example of 2 simultaneously recorded cells. This was directly activated, and this was an interneuron. It's exactly the 2 cells that I showed before. And you can see that the pyramidal cell spiked with relation to the input. So when there were deflections, this cell spiked, etc. But we can also see that the interneuron spiked very precisely with relation to the input. We can also say that the interneuron spiked more than the pyramidal cell at every instance. And when we quantify these things, we see exactly that. Okay, so first, when we compute the mean Wiener filter over a population of several hundred pyramidal cells and simultaneously recorded interneurons in many different experiments. So this is from, I don't know, 40 different sessions in 7 different mice. We can see that the pyramidal cells, the filters for the pyramidal cells are closer to the spikes than the filters for the interneurons. So the time lag for the spiking of the pyramidal cells is shorter by about 2 milliseconds. So this actually just shows that these interneurons are indirectly activated. There's another delay until they're activated. And this 1.6 milliseconds actually corresponds exactly to the mean delay of the cross-correlation peak. The surprising thing to us was this, that the pyramidal cells have a precision which is, in this case, it was 1.8 milliseconds. But this interneuron had a precision which was actually higher. Now you can say, okay, there are many spikes here, but if you remove the spikes which are less precise, you can actually get the same firing rates for all of these cells, but these cells would still remain more precise. So this was the main thing that was for me, initially, it was quite amazing that the indirectly activated cells are even more precise than the cells that we activate. So we looked at specific pairs, whether each specific presynaptic pyramidal cell is less precise than the simultaneously recorded interneuron. And we saw that in some cases, the interneuron is less precise than the directly activated pyramidal cell. But in the vast majority of the cases, there is an improvement, or there is higher precision for the cells that are indirectly activated. I will call them from now on the IDA INT, the indirectly activated interneuron. And when we quantify, yeah, just I will finish the sentence. Sure. When you quantify the improvement or the difference in the precision, it is this number. It's 1.6 milliseconds. Yes.
So by precision, you mean the reconstruction of the noise injected, right?
So by precision, I mean exactly this. The time that when you reconstruct the injected noise, the reconstruction deteriorates. So we jitter each spike, and we measure how much do we need to jitter the spikes in order for the reconstruction to start failing. This is exactly what I mean.
So, but it seems like your neurons are quite variable, right?
Very, very variable, yes.
So how can you, I mean, I don't understand how can you say, like when you show this paired test in the next slide, yeah, you can see that this precision goes from 1 to almost 10 milliseconds.
Sure.
So how do you explain that? Like these are all the same pyramidal neurons?
Yeah, there's a lot of variability. There's a million things that are not the same. That's the whole beauty of it, right? We record from various layers in the neocortex, in various brain states, for various durations, at various intensities of the stimulus, etc., etc., etc. There's a million sources of variability. That's the whole point. Despite this, that's what we see.
Do you hear me? Why don't you explain it simply by averaging? I mean, this indirectly activated are just averaging over many activated cells.
Sure. Yeah. That's the next thing. I'm not expecting. Yeah. Of course, that's a possible explanation, and I will in a second I will test this and I will show you that that's not the case. But that's an explanation. So far, I'm just reporting the results. Okay. So yes. So actually, Tali prompted this next slide, and that's exactly the question, right? So how can precision improve over a synaptic interface? So let's use the following terminology for a second. So what we see is that the IDA INT precision is higher than the DA INT, DA peer precision. So I would refer to this in what follows as error correction across the pyramidal cell to interneuron interface. And this can occur by several possible conceptual, at least, mechanisms. One of them is what one may call a labeled line mechanism, or a grandmother explanation kind of thing. So it would be a strong connection from a single precise pyramidal cell. So among the zoo of these imprecise and variable pyramidal cells, there, as you noted, Lilach, there are some which are at the left side of the distribution, and it could be that these guys are the ones which actually dictate the precision of the postsynaptic target. So that's one possible explanation. And another possible explanation is what Tali suggested, is that there is some averaging. So for that, I would call I would refer to this model as some population or an averaging response. And in broader terms, you can refer to this as accumulation of information from multiple pyramidal cells or multiple presynaptic cells. And this can have various flavors, linear summation or superlinear summation or coincidence detection, and we will distinguish between these things in a moment. So how can you test this? So what we do is we since we're recording extracellularly and we're recording many cells at the same time, so we infer the connectivity from the extracellular records, as I explained earlier. And what we do is we generate a spike transmission curve for every pair of cells. So this is one cell and this is another cell recorded simultaneously. This is the cross correlation between them. And we take the region from the peak backwards and forwards to the zero crossings after we subtract a deconvolved baseline. And we plot it here. So this is what we call the spike transmission curves, which basically shows at which time after a presynaptic, after this cell number 1 spiked, at which time is cell number 2 possible to spike. And the probability or the gain of this to happen is the integral under this curve. And that number we define as the spike transmission gain. Now, when we plot this for 1,000 pairs or so of presynaptic pyramidal cells to indirectly activated interneurons that responded precisely, we see that the distribution of these numbers is around very small numbers. So the median gain is about 100th of a spike. So it's very rare that a spike in a presynaptic cell would generate, actually generate a spike in a postsynaptic cell. It's virtually, it doesn't happen, at least in this data set. It does happen in other data sets, of course, but in the cortical data, it does not happen. So if we want to, so that's basically the tool that we can use to test these models. And I will just say the models once more. We considered 3 conceptual models. One is the labeled line model, in which the connectivity is 1-to-1. This has some examples also in freely moving or in vivo preparations. The sum population is basically a linear summation of the inputs, and the coincidence detection model is a superlinear summation of coincident spiking. And all of these have quite a bit of, I just mentioned a few. This is not a good list. It should be many more, because there's many examples for each of these modes of transmission in the neuroscience literature. So what do? We take the spike transmission curve, and we take the actual spike train of a presynaptic cell or cells. We convolve the spike train with this transmission function, and we generate a rate function, a single trial rate function. From this, we stochastically generate a spike train. And when we do this for the labeled line model, we get things like this. They're very sparse spiking. It basically doesn't drive any spiking, right? Because you saw this, the median is about 0.01 spikes per spike. So it's very sparse. And we don't get any neurons, we call these synthetic interneuron spike trains. So I would refer this as S-int. We don't get any S-int which are have an AC response, or actually can be reconstructed, can actually have a filter that is sufficiently statistically stable to support a reconstruction. We can do this for a host of spike trains. So if we have, let's say, 20 pyramidal cells that all of them were recorded at the same time with a single postsynaptic interneuron, and that postsynaptic interneuron had a precise or some precision, it had some reconstruction and some precision, we can reconstruct its, we can generate a prediction for its spike train or a synthetic int based on exactly what Tali suggested earlier, by taking all of the spike trains. Each spike train will be convolved by the specific data-dependent spike transmission curve with a specific gain. We sum all of these to get a single trial rate function. So it will be different for every trial according to the statistics of the various presynaptic cells. And we generate from this synthetic int spike trains. You can see that these guys are not as sparse. There's quite a few spikes there. They spike, but they are not precise. Yes, so this doesn't work either. When we use nonlinear coincidence detection, so we implemented various models for this multiplicative, exponential. It doesn't really matter. The results are really the same. Once you implement anything that accounts for the coincidence in the timing of the presynaptic cells, you get spiking, and you get a rate function which looks much more similar to the input. And in about half of the cases, we can actually get S-ints that have a post-synaptic, that have an AC response, and therefore we can measure the precision. So here we tried various scenarios. We tried a noiseless scenario with no background spiking, and then we tried a realistic scenario in which we complement the firing rate of the simulated int or synthetic int spike train to the actual firing rate of the observed spike train. For the coincidence detection model or type of model, we use several. The reconstruction works for about half of the cells. So it's not as good. We're still missing something, obviously. It's about 50% of the units, but this actually manages to reconstruct the precise or reconstructed spike trains. Now, once we saw this, we started thinking about why did we not manage to get a stable reconstruction for the other cells. So we actually looked at all the interneurons that were recorded and were indirectly activated. And we also looked at several other, maybe 400 neurons that are interneurons by statistical classification, but they did not respond to the white noise patterns. So they are also recorded simultaneously with the directly activated pyramidal cells, but they did not respond to the white noise. And we measured how many of the cells have at least one presynaptic peer or pyramidal cell that was presynaptic to it. And in the IDA INT group, almost all of them had, whereas in the others, only about half. The fraction of the simultaneously recorded pyramidal cells that were presynaptic to each IDA INT was several times larger for the interneurons that were actually responding, and the precision was better or higher or a shorter time scale when more presynaptic peers were recorded. So the take-home message for this is that when we record or we control or drive a larger fraction of the presynaptic population that drives a given neuron, we actually, this is somehow sufficient to actually drive that cell with a very short, with high time scale, which is actually considerably higher than the time scale of the presynaptic or the pyramidal cells that were driven. And this is actually also recapitulated in the coincidence detection model. So the error of the coincidence detection model is this is unsigned, yes, so it can be negative or positive. Error is smaller, and it's actually very small when the number of presynaptic pyramidal cells that were recorded is large. Okay. Okay, so I have about 11 more minutes, so I will make an interim summary. So this doesn't mean that I have still halfway to go. This is almost done. But let me just line out what I told you. What we said, what we built is a framework for defining temporal timescale or timescales in spike trains in relation to a fixed input. We define the precision, which is timing relative to a specific input, and the reliability timescale, which is timing with relation of one spike train with relation to another spike train under identical conditions. And we showed that these things can be dissociated. Specifically in the neurocortical peer-to-int interface, we found that int precision is higher than the precision of the presynaptic train spikes or cells which were directly activated, which we call or name as error correction, which is this phenomenon. And third, we found that the error correction is consistent with this type of model of nonlinear summation of coincident spikes. So I would like to emphasize that this nonlinear model is not just nonlinear amplification of every spike, it's specifically amplifying the coincidences, of course. Okay. So that was one axis, yes, the precision axis. But what about the reliability timescale axis that I took so much time in the beginning to show and try to convince you that they're dissociable? So this is another neuron from another animal in the neocortex that exhibited these kinds of spike trains over multiple representations. This is the precision. It's an indirectly activated interneuron. And when we apply the methodology of taking each spike train and convolving it with various Gaussians of various sigmas, we get this kind of a profile. So this specific interneuron, the indirectly activated interneuron, had a peak of the reliability or a peak or reliability timescale of about 4 milliseconds. And this is a relatively short timescale. But from this, I have nothing to say, right? Because I can't compare it to anything. I need to say, okay, how is this reliability compared to the pyramidal cells that drive it, as I did before? Now, it turns out that the pyramidal cells that drive it are all over the place. Some of them have a peak at short time scales and some at long time scales, and we couldn't make any order of this. Related to Lilach's question earlier, there we found nothing consistent. We couldn't make any order of this. But what we could ask, or what we were worried about, is whether this short time scale, or we were curious about, is whether this time scale is a property of the interneuron or of the fact that it's indirectly activated, whether it's a property of the transmission, the fact that it's indirectly activated, or whether it's simply all interneurons are like this. So what we did is we took another set of another 7 animals and we recorded in them interneurons while we directly activated them. Now these experiments are super low yield, but we managed to collect some sufficient number of examples to actually look at this. And these are the kinds of spike trains that you see. You can see that they are not as punctuate as the indirectly activated interneurons, but still the precision is the same. But what is evident is that the reliability profile is monotonously increasing. So the larger window of integration you use in the digital to analog conversion, the more similar these spike trains are. And this is a consistent property. So these are the reliability profiles for a bunch of all of the indirectly activated interneurons and all of the directly activated interneurons that we recorded in the neocortex from all of these animals. And these are the specific profiles looked at from above. And these are the timescale distributions. So you can see that the median timescale distribution for the directly activated interneurons is on the order of 100 milliseconds, whereas for the indirectly activated interneurons, it's an order of magnitude smaller. It's about 10 to 12 milliseconds. So now there's 2 more questions that I would like to ask or present to you what we found about. One is, so, okay, if this is so nice, so what's the cost of having an additional interface? So now I want to transmit a message. I always want to transmit a message, and I can just talk to this interneuron directly. The message that I'm playing now is the message of the optics, but it can be any peripheral input or any thought. So this is the one mode of transmission, and the other, so this would be a labeled line type of transmission. And the other one is to transmit the message to everybody, to all of these pyramidal cells, and then hope that they will manage to do some error correction when they average out or summate or coincidence detect or whatever. So what I showed you up to now is that this thing actually can work. And I also showed you that I can convey this message to the pyramidal cells and this. So now I would like to compare the two schemes of information transmission, the directly activated scheme or the two-layer transmission. Okay. So basically what I'm going to do is to compare the statistics of the indirectly activated int with the directly activated int. And specifically, what we found that is interesting, I think, is that there is a difference in the time delay. When you activate the cell directly, the time scale of the time lag is under 1 millisecond on average, or the median is about 1 millisecond. So it means that the cells start spiking very quickly after a transient in the white noise, whereas the indirectly activated interneurons, as I showed you before, take more time. So from this event to this event, several things happen, right? The cell, after the cell spikes, it has to, the potentials have to travel in the axon, through the synapses, through the dendrites, to the post-synaptic cell, to integrate there, or whatever, and generate a spike, and only then we see this. So these 3.2 milliseconds is the total delay of all of these things. It's not simply the transmission. The transmission and the spike generation of the post-synaptic cell. But what we found is that the precision is more or less the same in the two schemes. So you don't lose precision by adding these two layers. And what you can gain is actually robustness. So we can't really test this by lesioning individual cells. We did not, maybe we can, but we did not test this by actually lesioning individual cells. But what we did do is lesion cells in the coincidence detection model. So this is the median precision for the coincidence detection model. And this is the precision for the actual data. And what we did is we randomly chose a pyramidal cell for each synthetic int, and we removed it. And we, of course, the precision deteriorated a little bit, but we didn't lose cells, and the precision did not deteriorate drastically. So, of course, this is an incomplete answer to robustness, but this is what we did. And the final thing that I will show you is the architecture. So what I showed you up to this point is everything was from the neocortex of Tel Aviv mice, but this could be forget the Tel Aviv, yes. It could be specific to a network that has a rather extensive or specific connectivity that the neocortex displays. So, but the motif of pyramidal cells to interneurons also exists in many other brain regions. So we repeated all of these experiments, most of them in the same mice. So we put the probe in the neocortex, and then we translated gradually also to CA1. So we have this opportunity to also test the same thing in CA1, and we repeated all of these experiments in CA1. All of the neurons are from the pyramidal cell layer specifically. So these are two different types of mice, the ones in which we activate the pyramidal cells and we look at the indirectly activated int, and the ones in which we activate the interneurons directly. So we call them DA int. You can see clearly that the DA int have a peak. Sorry, the IDA int, the indirectly activated cells have a peak, whereas the directly activated pyramidal cell interneurons, and also here it's actually a bit clearer, the directly activated pyramidal cells have a monotonously increasing reliability profile, so the reliability timescale is longer. And these are the overall statistics. So the reliability timescale of the directly activated cells, both the pyramidal cells and the interneurons in CA1, is about an order of magnitude longer than the reliability timescale for the indirectly activated cells. and the temporal precision is similar for all of the types, or it's not significantly different. This is what we should say for the different types of interneurons, but the indirectly activated interneurons improve the precision relative to the presynaptic pyramidal cells that are directly activated. So the error correction is maintained. So yeah, this is just in time to summarize. So I will again tell you what I told you. The pyramidal cell to interneuron interface exhibits error correction. So I don't know if this is a general thing that any interface, any feed-forward excitatory interface does. We did not test this in pyramidal cells to pyramidal cells, but in both structures that we tested exhaustively, the pyramidal cell to interneuron interface exhibited error correction. This was consistent with this coincidence detection model of nonlinear summation at the specific time of coincident spikes. And the third thing that we saw is that the synaptically activated cells exhibit particularly short reliability timescales, an order of magnitude faster than the directly activated interneurons, which is actually consistent with what people sometimes call temporal coding as opposed to rate coding. And these results were similar for both neocortex and CA1. So this is the two-dimensional maps that I made at the beginning with the synthetic data, and you can see where these actual data fall. So there's many dots here, many different colors, but the crosses show you the pyramidal cells in the neocortex and in CA1, and which have relatively high or long reliability profile and some precision, and the postsynaptic interneurons in both regions have a shorter reliability profile and a higher precision. Okay, so that's all I wanted to share with you.
Thank you, Eran. So we have time for questions, so feel free to open your mic and ask questions.
I have a question again. Have you tried injecting light that is not white noise, like something that is more structured and
Sure, yeah, we do this all the time. Yeah.
But do you see the
question?
same effect? Like, do you see the same thing?
Yeah, you can't really ask these kinds of questions when the input is narrowband. So we, of course, we inject various pulses and sinusoids and chirps, linear chirps. We use a million types of different. We use some feedback on the system. So we do a lot of different types of stimuli, but you can't really ask this question with structured stimuli. You need something that spans the space of timescales sufficiently.
Oh, I see.
Okay.
But then the question is how relevant it is to like natural world, you know, because the world is consistent of, you know, very structured.
Yeah. No. Sure, it's nothing real. This is an experiment. That's the whole. I agree completely. I don't propose that any cell in the brain receives the input that we gave it. This is a synthetic experiment. I'm just saying that when you give that kind of an input, you can measure things like precision and reliability timescale, and when you do that, you get these results. You get then you can reveal this thing. If I would do this with 10 hertz sine waves, I would, or 1 millisecond pulses, I would never be able to ask this question.
I see. Okay.
So maybe I can ask a small question. So you mentioned that you completely ignore cortical states, right? But most probably those cortical states have very dramatic effect on these functions. So do you have any plans or idea? What do you think this is might be happening or operating?
Yeah. Yeah, well, you can see that every time. Here, you can see here, look at this. We deliberately left timing outside of the simulation. You can see here that there are more spikes here than in the preceding and following. Yes, and also during the stimuli, there is something different in these two spike ranges or three than others. The spike ranges are spaced out, so two spike ranges of 1 second can actually be taken at about a minute interval. So this may correspond to REM sleep or something like this. So yes, these results are averaged over all sources of variability, including very many cortical states. We could analyze these things as a function of cortical states, and I'm pretty sure that we will find something. We didn't do that.
So I have a question, which is just to follow up on the last two questions. So first of all, you're using a stationary signal here. I mean, it's a stationary non-white noise, if I understand correctly. But what you would like to do is some sort of a stationary Markov chain of different colored stochastic signals, which will really give you both meaningful signals and something like cortical states. And it can be that the precision will be much higher than, no? I mean, this nonlinear error correction mechanism is specific to, obviously you can see how it works, but I think it will work much better if you have a chain of repeated states, like a Markov chain of stationary but switched states. Have you tried this?
No. This experiments, sure, yeah. First of all, I agree, and this is a great idea. The problem is that these experiments are super low yield. So to gather 100 cells that actually respond to the white noise, you need to record 3,000 cells or so. So this is recordings over years. We do vary many things, as I said. We vary the intensity of the stimulus, the position of the light source. There's a million things that do vary over these experiments. So yes, I think these are great things to do in the next stage. If you feel that these results are convincing, then these are things that we could base the next stage of experiments on. But first I need to convince people that these things are sound. So that's basically the input that I would like to receive. Whether you see any flaws here or any things that we miss.
Thank you.
Any more questions? So I think we'll conclude here, and we'll thank Eran for this interesting talk, and we hope to see you everyone next week.
תודה, ערן.
תודה רבה.